Now we will discuss the topic that is the morphological changes, which we have read earlier that when the spermatid is going to form the spermatozoa, mature spermatozoa, there are some morphological changes taking place in spermatid that time. And these morphological changes are taking place in nucleus, Golgi apparatus, centriole, mitochondria, cytoplasm and plasma membrane. Step by step we will discuss all the organelles in which the changing is taking place and what kind of changes are taking place. First we are having nucleus. So what change is going to take place in nucleus? That is, the nucleus in spermatids is going to elongate and condensation is taking place. Condensation means removal of water is taking place. Shape of the nucleus is changing from circular to oval. This we will discuss the how shape is changing from circular to oval. And next is chromosomes are compactly arranged. When the chromosomes in the nucleus are compactly arranged and there is no kind of transcription, means there is no mRNA formed and there is no protein formed. So how do nucleus is going to form the proteins, or how do it is getting the proteins, that we will discuss now. In chromosomes, we are knowing in normal eukaryotic cells the DNA is associated with normal histone and non-histone proteins, but in case of sperms, during the transformation, these histones and non-histone proteins are replaced by proteamines. These proteamines are having cysteine residues, and these cysteine residues have the sulfur group along with them. And this is the structure of chromosome in which two cysteine groups are present and they are having the sulfur groups, and these two sulfur groups of the cysteine groups are joined by disulfide bond. And this sulfur groups, when they are joined by the disulfide bonds, this make the structure of chromosome compactly arranged or coiled so that it becomes transcriptionally inactive and no mRNA is formed and there is no formation of protein. And now how do it is getting proteins? It is getting the proteins from the mRNA which is present already in the nucleus, that is providing it proteins for the further processing. Now we will discuss the change that is taking place in the Golgi apparatus. We have discussed that nucleus is formed after the morphological changes. So imagine the place where the nucleus is present is here. Now we will discuss the Golgi apparatus. This is Golgi apparatus. During the morphological changes, this Golgi apparatus is secreting some proacrosomal vesicles. These proacrosomal vesicles are containing some secretions that are hydrolytic enzymes. Which proacrosomal vesicles are secreted by Golgi apparatus? These all vesicles join together and form this part above the nucleus. This part. It forms this part above the nucleus. And these secretions which are present in the proacrosomal vesicles, these dotted secretions which are present in proacrosomal vesicles, these secretions join and form here acrosomal granule. This dark part you are seeing, this is the acrosomal granule which is formed by the combining of all the hydrolytic enzymes. These are the secretions which are present in proacrosomal vesicle. This part with acrosomal granules and acrosomal vesicles, this all part is known as acrosome, and it is present at the top of the nucleus and it forms a cap-like structure above the nucleus. And what is this? Manchette, we call it manchette. It is the group of microtubules which is present at the sides of acrosome, at the posterior end of the acrosome around the nucleus. We have discussed the shape of the nucleus is changing from circular to oval. So how it is changing, we will discuss now. This manchette is the group of microtubules. When it compresses the nucleus. So the shape of nucleus is changing from circular to oval. So its role is in the changing of the shape of the nucleus. So we have discussed that acrosome is the cap-like structure present on the top of the nucleus. So So we have discussed earlier the morphological changes that are taking place in nucleus and in Golgi apparatus. Now we will see the changes that are taking place in centriole, in plasma membrane, mitochondria and cytoplasm. So we have seen this nucleus is formed and a cap-like structure, that is acrosome, is formed at the above of the nucleus. This is the cytoplasm in which all the morphological changes are taking place. This is the Golgi apparatus and these are the centrioles. Now we are knowing that Golgi apparatus is no use, it have done its work. That is, it has released those proacrosomal vesicles which have made this acrosome. Now there is no work of Golgi apparatus, so it will move at the posterior end of the cytoplasm. And these centrioles will also move at the posterior end of the nucleus, in which one centriole will be known as proximal centriole and other will be known as distal centriole. This proximal centriole will make the neck part down the nucleus and this distal centriole will make the flagellum part that will reach the tail part. So now we will see here. This is the nucleus. It has acrosome at the top, that is the cap-like structure. And this cap-like structure or the acrosome around the nucleus is known as prenuclear cap. And we are knowing, I have told you earlier, that the acrosome is having manchette around it which covers the nucleus or compresses the nucleus. That acrosome around the nucleus is known as postnuclear cap or the manchette part. This is the head part in which there are the acrosome and the nucleus. So we have discussed here that proximal centriole is going to form the neck part. So here this neck part is formed by proximal centriole and this distal centriole is now going to form this flagellum part. So this flagellum which is arising here and reaching the tail part, this is formed by the distal centriole. So this is the middle piece and this is the tail part. So how we will differentiate this middle piece and the tail part? This middle piece is having this mitochondria around it which will provide it energy, and this is the tail part where there is no mitochondria, there is only the flagellum part. So what changes are now occurring in the plasma membrane and cytoplasm? Cytoplasm will now cover the whole part of the sperm. It will cover the nucleus, the neck part, middle piece and the tail part as the sperm is going and forming. And what the changes taking place in the plasma membrane? Plasma membrane will now undergo the differentiation in which some proteins will attach with the plasma membrane which will help to recognize the sperm, its egg or the ovum during the fertilization.